Hello again, everyone. So I'd like to continue our discussion on the chemistry of nucleic acids and focusing again primarily on DNA. We've talked about the composition of the nucleotides, how they can be strung together into these long polymeric chains to create either a DNA or an RNA molecule. And then we talked specifically about the Watson-Crick model for double-stranded DNA, which represents the predominant form of DNA on the planet, and utilized as a source of genetic information in the cells of most life forms that we're aware of. So I'd like to continue that discussion a little bit with regard to DNA structure, and then some of the important measurable biochemical properties that can be used in some sort of a practical way in the laboratory. So first of all, going back to the bDNA model for DNA of Watson and Crick, again, remember that model relied very heavily on data generated by Rosalind Franklin. And so she generated these X-ray diffraction patterns. So what she was actually able to do, and what I described to you, is that she described two different diffraction patterns depending on how she prepared her fibers of purified DNA, using different methodologies to dry down from a liquid solution those fibrous forms of DNA. And again, using different relative humidities and so on. So she was actually able to generate two different diffraction patterns, what she called aDNA and bDNA. So again, what that suggested is that DNA can exist in multiple forms, and actually that's the reality. So some of these forms are artificially created in a laboratory, but some of them exist in nature, have some sort of biological significance or biological relevance. So I just want to talk briefly about, these alternative forms for DNA and some of the roles for these alternative forms that that we're aware of, or that we have some understanding of. So again, the aDNA, as originally described by Rosalind Franklin, has been studied significantly beyond that point in time, and its structure is fairly well understood. As it says here, it's slightly more compact than bDNA. So that what that makes for is it's a cylindrical molecule, two single strands of DNA held together by the same types of base pairs described for bDNA by Watson and Crick. There's a helical twist to the molecule and so on, but it's a more compact molecule, a more compact cylinder, and therefore it has a larger diameter than the 20 angstroms of bDNA. zDNA is another very well characterized form of DNA as well. And what's fundamentally different between zDNA and bDNA, again, zDNA is double-stranded. The two strands run anti-parallel. The two strands are held together by the same types of base pairing that you see in bDNA. But what's fundamentally different about zDNA is that it's a left-handed helical twist as opposed to a right-handed helix that you see in bDNA and that you see in aDNA. So we'll talk a little bit, we'll do a comparison in just a second. I do want to point out there are many other forms of DNA that have been created, again, initially artificially in the laboratory. So there's a C form, there's a D form, an E form, and here they describe these different forms as being less compact than bDNA. So in other words, the diameter is going to be thinner than what you see in bDNA. There's also hDNA, and we'll talk just briefly about that. hDNA is actually triple helical. So DNA, we already know it can exist in a single-stranded configuration. It can exist in a double-stranded configuration, but it can also exist in a triple-stranded configuration as well, a triple helical molecule. Okay, so what I've given you down at the bottom of this slide here is a link to a video in YouTube where they talk about, it's about a 10-minute video if you're interested in these different forms of DNA and biological relevance and so on. They go into a little bit more detail than what I'm going to give you here in this lecture. And it's a really good presentation in my mind, at least. Okay, so here's just a ball stick model for A, Z, and B DNA. So we've talked about the B DNA already. The red ribbon represents the outside of the cylinder, the double-stranded helical cylinder with that right-handed twist to it. The red ribbon represents the sugar phosphate backbone. And then what you see interior to the cylinder again are the base pairs themselves, AT and GC. And then you can also see very clearly in this representation, the major and the minor grooves that allow for access to that informational component of the molecule. Okay, so you can contrast that with zDNA and aDNA, and they look different. So you can see clearly that the aDNA has a much wider diameter than either B or Z DNA. And because of the nature, the alteration to the twisting of the molecule, again, remember, it's still double-stranded. There's two strands are anti-parallel, same types of base pairing as in bDNA, but the nature of the twist to the molecule is different. So that alters significantly the nature of the grooves. And again, remember, it's the grooves that allow access to the informational content of the molecule. So then here's zDNA, and again, this is double-stranded, anti-parallel strands, same types of base pairing, but it has a left-handed helical twist to it. So it makes the molecule thinner than bDNA and certainly thinner than aDNA. And again, it alters significantly the nature of the backbone to the molecule. These red ribbons of sugar alternating sugar and phosphate. So there's sort of a zigzag pattern in zDNA that you don't see. You see more of a smooth ribbon for bDNA and aDNA for those alternating sugars and phosphates. So that's where the name derives, the zigzag pattern of the alternating sugars and phosphates in zDNA. And you can see again, because of the left-handed helical twist and so on, is that you alter significantly the nature of the grooves and again, alter significantly the way that the informational content of the molecule can be accessed in the cell. So this more compact nature to aDNA, the only thing I want to point out here is the idea is that in bDNA, when you have a base pair, it's perpendicular. Remember, it's initially I described it as a ladder. You create a ladder. You have the rungs or the steps of the ladder, and they're essentially horizontal, perpendicular 90 degrees to the backbone. But you can see in this more tightly coiled, more compact molecule of aDNA is that those base pairs, again, are at an angle. So again, that leads to, again, the alteration of the nature of these grooves and an alteration to the means by which, you can access the informational content of the molecule. So here's zDNA, again, with the left-handed helical twist. And again, just to point out is that the way that the base pairs form in the cylinder, again, are very different from bDNA. They're at an angle again, as opposed to being 90 degrees to the backbone, to the sugar phosphate backbones. So again, a little bit different, and again, that comes about because of the altered pattern of twisting that occurs in zDNA relative to bDNA. So there's two tables here. You can look these over on your own, but just giving various differences for a variety of characteristics when you compare A, B, and zDNA. I guess the main things I want to point out is that, so again, as I said, aDNA is has the largest diameter. So you can see, so it's broad. about 25 angstroms. Remember, we said for bDNA, it's about 20 angstroms. I should point out, here they say, 23.7. So, all of the various parameters that Watson and Crick described in their original model, they've subsequently been modified to some extent, because of, increased study of the molecule by other chemists and biochemists and so on. So coming up with more precise measures. So that 20 angstrom diameter, 20 to 24, somewhere in that range. Then you can see zDNA in terms of, the diameter is the smallest relative to either A or B DNA. Again, we talked about the nature of the helical twist, right-handed for A and B, left-handed for Z. Because of the nature of the twisting is, what we said, for bDNA, there's 3.6 angstroms between two adjacent steps in the ladder. So each base pair turns 36 degrees relative to any adjacent base pair. So a complete 360-degree turn encompasses about 10 base pairs. So that's reflected here. So you can see there's roughly 11 base pairs per 360-degree turn of aDNA because of that altered twisting pattern. And there's about 12 base pairs per 360-degree turn in zDNA. And again, those altered twisting patterns alter the nature of the grooves for either A, B, or Z relative to each other. So again, for bDNA, wide and deep for the major groove, narrow and deep for the minor groove. And again, deep in the sense that you can read all of the information within the molecule, the sequence of bases within the nucleotides. So those are notches, those are grooves that are deeply cut into the molecule. So you can see, again, they're altered in aDNA. Here they're narrow, a narrow major groove, a broad and shallow minor groove for aDNA. And the grooves are either flat or very narrow and deep in zDNA. So fundamentally different configurations for, very similarly constructed DNA molecules. So, one of the questions is, well, what's the significance of that? And does it have a biological significance or biological relevance? So we know that, the majority of DNA that exists is in the B form, again, contained in all the chromosomes of all the cells of living organisms. That's the biologically relevant or biologically significant form of DNA, serving the role as the carrier of genetic information. What about aDNA and what about zDNA? So aDNA actually is pretty rare. It was, again, initially described or created artificially under laboratory conditions. And as far as I'm aware, there's only one example in a sporulating bacterium. When the spores form and it goes dormant, is that the DNA can revert to an A configuration in that dormant state. Okay, so it's pretty rare for double-stranded DNA to be in an A configuration. However, there is a biologically relevant form of A nucleic acid. So actually, double-stranded RNA is predominantly in an A configuration. So you can have double-stranded DNA molecules. The nature of the base pairs would be A and U and G and C. The two strands would run anti-parallel to each other, and they can base pair, and they can assume a helical twist, and so on. So because of that extra OH group, that 2' carbon of the sugar of the nucleotides, is that the helical twist is such that you create a nucleic acid or, double-stranded RNA. In addition, you also see in DNA, RNA hybrid molecules. So DNA, a single strand of DNA can stably base pair with a single strand of RNA, and it can assume a helical twist. And when it does, the form of the twisting is such that you create a nucleic acid or DNA RNA hybrid. So that's biologically relevant in the sense that those types of hybrid nucleic acids are created in vivo, and it's during the process of gene expression during a transcriptional event. Okay, so aDNA, not aDNA, but DNA RNA hybrids assume this A configuration, so there is biological relevance to it. And that's important for the transcriptional machinery, the proteins that are carrying out the transcriptional event. So what about zDNA? So again, zDNA, I think it was in the late 1960s that it was first artificially created under laboratory conditions. And so then the question is, does it exist, under, biologically relevant conditions in a living cell? And actually it does. So again, remember, in a chromosome, say a human chromosome running from end to end, within that chromosome is one long molecule of double-stranded B DNA. But there are subportions of that molecule that can, so it's not uniformly B DNA along its entire length. There are subregions of the DNA molecule that actually can twist in the Z form. So it does exist in living systems, zDNA, but as sections or portions of a DNA molecule. And the question is as well, what does that serve any sort of function or any sort of role? Well, there's plenty of evidence to suggest that it plays a regulatory role, determining whether or not a gene is active or not. So in other words, zDNA occurs predominantly in the region of a gene that is important for initiating a transcriptional event or gene expression. So we'll talk more about that later. But the idea is that it seems to have a regulatory role in determining whether or not a particular gene is allowed to express its information or not within the cell. So one last form, as I said, for DNA that, again, first created artificially, but also shown to be, biologically relevant. In other words, it can exist in living systems, is hDNA or triple helical DNA. And I've just given you different representations of, what would be considered triple helical DNA here. This is probably the best diagram over to the right. So it's essentially, bDNA, the two green ribbons, and then you have that major groove within which you can wrap a third single strand of DNA. And you can get, base pair interactions, hydrogen bonds forming between all three bases within that triple helical region to stabilize the complex. So the question is, again, is there biological relevance? Well, actually, there is biological relevance, and it's a relevance that is of significance to a lot of pharmaceutical companies trying to develop DNA-based drugs. So the idea is that, say a diseased state or a disease condition where, gene expression, say cancer, or gene expression patterns have gone haywire, or there's an infection by a bacterium or a virus, whatever the disease state, there's, expression of genes involved in creating that disease state. And so the idea is that can you reverse that condition if you shut down the activity of certain genes, whether it's the cancer-causing genes endogenous to an individual's genome, whether it's a viral gene, whether it's a bacterial gene, whatever gene you're targeting. Well, presumably what you should be able to do is create a strand of DNA that can recognize particular genes and then interact with that double-stranded gene to create this triple-stranded helical molecule. And in doing so, again, what's happening is by adding that third strand of DNA to the gene is that you limit access to the informational content of the molecule again. And therefore, you would decrease the overall activity of that gene. You'd shut it down or turn it off. And again, if these are genes that are involved in contributing to the disease state, then presumably you could reverse that state. So there's all kinds of efforts nowadays to develop these DNA-based drugs to treat a whole variety of different conditions. Okay, I don't want to complete this discussion without at least briefly at this point mentioning RNA. So we've talked about DNA, the chemistry and the Watson-Crick model and these different configurations and so on. So there is this other type of nucleic acid, the RNA. We've talked about, again, the nature of the nucleotides, what the RNA molecules look like when polymerized and when those nucleotides are polymerized and so on. So I just want to mention briefly here is that, RNA in a living system plays a central role in expression of the information that's contained within a gene, within a double-stranded DNA molecule. And so, in other words, the ability to express the information contained in that gene requires the involvement of different types of RNA molecules. There are some RNA molecules that can be regulatory. In other words, they can, again, similar to what I just described, the hDNA, the triple helical, you can have RNA molecules that interact with genes or interact with other RNA molecules that regulate activity of or levels of gene expression. So they can be regulatory in nature, but in addition, these RNA molecules are key components to the actual expression machinery of the cell. So the end result of gene expression is to take, nucleotide sequence or base pair sequence information from a DNA molecule and use that information to create a protein. So again, the different types of RNA are integral to that process of gene expression. And there's three predominant, these aren't the only forms of RNA in a cell, but these are the predominant forms. There's messenger RNA, mRNA, ribosomal RNA, rRNA, and then transfer RNA or tRNA. So these are RNA, they're all the same. We'll talk more about them when we talk about transcription and translation, the actual process of gene expression. But for now, they're all RNA molecules, built from the same 4 subunits, strung together through the same 3' to 5' phosphodiester linkage. And they all act predominantly in a single-stranded configuration. So very different from DNA. Again, DNA, as I said, is predominantly in a double-stranded form. The bDNA is described by Watson and Crick. But these RNAs carry out their functions primarily in a single-stranded configuration. So again, I've shown you this multiple times now, but here's the RNA. And again, the fundamental difference between RNA and DNA is just very subtle. So there's this 2' OH on the sugar, ribose sugar as opposed to deoxyribose. And then there's that very subtle difference between one of the 4 nitrogenous bases, one of the pyrimidines, thymine, which is a methylated form of uracil, and it's uracil that you find in the nucleotides of an RNA molecule. So again, in terms of functionality, as I said, these single-stranded RNAs contribute to the machinery that allows for protein production in a cell. So rRNAs, ribosomal RNAs, are components of the ribosomes, the actual protein synthesis machinery within the cell. tRNAs, transfer RNAs, carry the amino acids, the individual building blocks for protein assembly in the cell, carry those amino acids to the ribosome that then lead to, protein assembly. And how is the order of those amino acids determined? How does the ribosome know, the sequence of amino acids to generate during this process of synthesis? That information is provided by mRNA. So mRNA is an RNA molecule that is produced off of a protein encoding gene. And it's the mRNA that carries information that tells the ribosome what order of amino acids to create during the process of protein assembly. Okay, so we'll talk more about all of that later on in a future lecture. Okay, so the last thing I want to talk about with regard to the nucleic acids, and again, this will be predominantly, focusing on DNA, is to describe some measurable biochemical properties of these nucleic acids that have practical utility in a laboratory setting. So again, as I've said, modern genetics is molecular, or predominantly molecular. Manipulating DNA, manipulating RNA in a laboratory setting to address various questions about functionality, structure, function, and so on. So there are certain relatively easy measurable biochemical properties that that are useful in this setting. So the first one has to do with the ability to absorb light. So again, there's many different wavelengths of light that exist, and based on the atomic structure of a particular compound or material, there are going to be particular wavelengths of light that are maximally absorbed. So that's certainly true for nucleic acids, both DNA and RNA. And both DNA and RNA exhibit maximal absorbance of a particular wavelength of light at 260 nanometers. And that happens to be the wavelength of light that falls within the UV light range. So what is the property of the nucleic acids that confers this property? Well, it has to do with the nature, the chemistry of the bases. So again, as I've described, the purines and pyrimidines, they're both ring-like structures, and the rings consisting of carbon and nitrogen atoms. So that particular atomic structure or configuration confers this particular property in terms of absorbance of a particular maximum absorbance of a particular wavelength of light. So, other compounds, as I said, based on their chemical makeup, their atomic structure and so on, they'll exhibit different wavelengths of light where they exhibit this maximal absorbance. So again, proteins assembled from amino acids, the nature of the amino acids are such that they actually absorb maximally at 280 nanometers, whereas again, the nucleic acids absorb maximally at 260 nanometers. So what's the value of being able to measure this property, this absorbance characteristic? So actually what you can do is you can use that measured value as a way to assess the purity of a DNA sample that you have purified in the laboratory, for whatever experiment you're going to perform. So it allows you to determine the purity of the sample, whether you have contaminants in there or not, or if you have pure nucleic acid. And then in addition, it also allows you to quantify the amount of nucleic acid that you have in the sample. There's a proportionality between the amount of absorption at 260 nanometers and the actual amount of DNA or RNA that you have in your sample. So those are the practical ways that this particular property is utilized in the laboratory. So here's a more complete absorbance spectrum for double-stranded DNA, and the blue line represents double-stranded DNA. So again, along an axis of different wavelengths of light from about 180 nanometers to 300, here's 260, and that's where you see, again, the nucleic acid, in this case, double-stranded DNA, exhibits maximal absorbance. So again, based on that measurement, you can then quantify the amount of DNA that you have in your sample, and you can also, based on the overall nature of this curve, is that you can determine the purity of your DNA sample. Okay, so the other thing that's being demonstrated in this diagram here is the idea that, again, remember, it's the nitrogenous bases that are absorbing the UV light that give you this particular absorbance reading. So, but again, in a double-stranded DNA molecule, or native DNA, as they refer to it here, is that those bases are interior to the molecule. So they're less exposed and therefore have less access to absorb any available UV light. However, if you break the double-stranded DNA molecule, in other words, convert it from the double-stranded form to the single-stranded form, then what you see is a change in the absorbance reading of that same DNA sample. Now, if the DNA, the original double-stranded DNA that gave you this first curve, the blue curve, if that DNA was converted into single-stranded DNA, it would give you this second curve. So in other words, the absorbance reading increases. And that makes sense because when you denature the DNA, you convert it from the double-stranded to the single-stranded form, you expose the bases. So they're more readily accessible or available, or to absorb any available UV light. So the absorbance reading goes up. Okay, so again, how do you denature the DNA? Well, all you have to do is break the hydrogen bonds that are holding the two strands together in the double-stranded configuration. So you can do that with chemicals, you can do that with heat. We'll talk a little bit more about that later. So this increase in absorbance when you denature double-stranded DNA or convert it from a double-stranded form to a single-stranded form, this is what's called a hyperchromic effect. Okay. And again, as you see, when you take measurements of that hyperchromic effect, there's information that can be derived from these types of denaturation analyses. Okay, the second measurable property for nucleic acids, and certainly not just for nucleic acids, is you can measure, there are ways, laboratory techniques that allow you to measure the density of a particular molecule or substance, an organelle or whatever, cells, whatever. So by measuring the density of nucleic acids, and in this case we're going to focus specifically on DNA, is that you can derive useful information about the structural organization, the nucleotide composition of a DNA sample. So how do you go about measuring the density of a DNA molecule? Well, it's through a process that's referred to as a density gradient centrifugation or sedimentation equilibrium centrifugation. So basically what's involved in this process is the following. So down here at the bottom of this diagram here, you've got a culture of bacteria. So again, what you can do is you can harvest those cells through centrifugation, just like Avery McLeod-McCarty. You can purify the DNA component of those cells. Okay? And once you've purified the DNA component of the cells, you can then use that to measure density. So how do you do that? Well, typically what's done is, again, it's a process of centrifugation. And the centrifugation is such that you're using a liquid material, liquid substance of a heavy metal salt, cesium chloride. And by centrifuging a solution of cesium chloride, what you do is what you will do in that centrifuge tube is establish a density gradient. So when you apply your DNA sample to the solution of cesium chloride, followed by centrifugation, you will have established a density gradient among that solution of cesium chloride, high density at the bottom and low density at the top. And then what will happen is through sedimentation equilibrium analysis is that the DNA sample that you've loaded to that solution will band out or will migrate to a position within the gradient that is equal to its own density. So in this case, we've added DNA from this bacterial culture. And what you see is that after the centrifugation, two bands of DNA arise, meaning that in the original bacterial cells, there must be two different populations of DNA molecules, one of which has a higher density than the other population. And so you can measure, as I said, you can measure that density difference. Because the DNA, the bands that you see in that gradient are the position within the gradient is equal to the density, the cesium chloride density is equal to the density of the DNA within that band. So how do you determine the density of the DNA? Well, the first thing you do is you fractionate your gradient. So you do your centrifugation, you get your two regions of banding. I should say at this point, you can't see the DNA. Okay, so how do you see the DNA? Well, again, that's where you use the absorbance reading at 260 nanometers. You take an absorbance reading. So we'll get to that in a second. So in any event, the first thing you do is, or at least the old way of doing this, is you just poke a hole in the bottom of the tube, the centrifuge tube, and you collect fractions. So in tube 1, you might collect the first 3 drops from that gradient, and you collect the next 3 in tube 2, the next 3 in tube 3, and so on. All the way through, in this case, 11 different tubes, collecting 3 drops at a time in each of these 11 tubes to fractionate the gradient. So again, remember the bottom of the tube is in tube 1, and the top of the gradient is in tube 11. So what you can do is there's instrumentation in the laboratory, a densitometer that'll measure the density of cesium chloride in each of these fractions. So you then know the density of cesium chloride in each of those 11 fractions. So then you have to find which of those 11 tubes contain DNA. So how do you find DNA? Again, you take an absorbance reading at 260 nanometers. And whenever you get a peak absorbance above 0, you know there's DNA in that tube. So that's true for fraction number 6 here and fraction number 9. So again, we've already measured the density of the cesium chloride, so now we know the density of DNA in those 2 different populations from the original gradient. Okay, so what's the value of measuring the density of this of DNA? So as I said earlier, what it allows you to do is it provides sort of crude information in terms of the basic makeup of that DNA molecule, basic information with regard to the nucleotide composition or the base composition. So the idea is, remember we've discussed previously as part of the Watson-Crick model, is that a GC base pair is held together by 3 hydrogen bonds, and an AT base pair is held together by 2 hydrogen bonds. So a GC base pair is a little bit more dense than an AT base pair because it's held together a little bit more tightly. So I don't think you can actually measure that density difference for a single base pair, but in a long DNA molecule, tens of thousands, hundreds of thousands, millions of base pairs in total length, you can detect a difference with regard to the relative content of AT base pairs to GC base pairs. So here's the relationship of density to percentage of GC base pairs. You can see it's a linear or a direct proportionality in terms of this relationship between GC base pair content and density of the DNA molecule. So again, DNA purified from many different sources, from bacteriophage to bacteria to Neurospora crassa. And you can see that the density of these different DNA molecules align very well with this, this linear graph here. So again, what happens is that as the GC content of the DNA increases, there's a direct proportional increase in the density of the DNA molecule. So once you know the GC content of the DNA, you also know the AT content of the DNA, right? Because those are the only two types of base pairs that exist within the molecule. So why is this important? Well, it's important, you know, we're still, we haven't discovered all the different life forms that exist on this planet. You know, they're finding new life all the time. Primarily, you know, microbial or bacterial, viral, and, you know, more primitive life, but, you know, they still want to discover it and characterize it and so on. So what's one of the first things you want to characterize nowadays? You want to characterize the nature of the genetic content of that organism. So one of the first things you can do is you can measure the density. You discover a new life form, a new bacterium. You know, if you can get it to grow in culture, you get enough DNA, you can measure density, and you come to this graph. You know, once you've measured the density, you can read over and get an estimate of the relative percentage of GC base pairs in that organism's genome. And again, once you know the GC content, then you know the AT content. So it's very basic descriptive information in terms of the nucleotide composition of the genetic material of all forms of life. Okay, so there's one other measurable characteristic I want to describe, and it essentially does the same thing. So this is the idea, we talked earlier about this hyperchromic effect. So here's a double-stranded DNA molecule to the left. Okay, so that double-stranded DNA molecule can be denatured. In other words, it can be converted from the double-stranded form to the single-stranded form simply by breaking those hydrogen bonds that hold the two strands together. So how do you do that? You know, you can do it chemically. There's different chemicals that'll break those hydrogen bonds. Or you can use temperature. You can use heat. As you increase the temperature of the sample of DNA, the hydrogen bonds start to break and the molecule denatures and eventually is converted into a single-stranded form. You can follow the progression of that reaction, of this process of denaturation, again by using the absorbance reading at 260 nanometers because of the hyperchromic effect that I described earlier. As the DNA denatures, the absorbance reading increases. And eventually it'll level off when the DNA is fully single-stranded. I just want to mention here that you can actually follow the reverse reaction. This is called renaturation. So in other words, once you've denatured the DNA, you can cool the sample down and you can allow it to convert back to the double-stranded form. And again, the reformation of the double-stranded form is going to depend on proper base pairing. And you can measure the rate of that reaction, the rate of renaturation. And actually, that process provides information about structural organization of organisms' genomes. Okay, we're not going to talk about that. We're just going to talk about this process of denaturation. So again, this process of denaturation is going to reveal something about the overall nucleotide composition of the original double-stranded DNA molecule. So here's a melting curve for DNA. And again, as I said, you follow this reaction by taking an absorbance reading at 260 nanometers. So here's temperature on one axis, absorbance on the other. Low temperature, 70 degrees, the DNA will be in a double-stranded form. So that's your starting point, time zero. And you start to heat it up. Once you get to a certain temperature, the DNA starts to fall apart. You start to disrupt those hydrogen bonds. So the DNA starts to denature. And you enter into this transition zone or this transition range. as the DNA is being denatured. 10% at the bottom, 50% in the middle, 90% at the top, and eventually it's 100% denatured as the temperature reaches a certain point, in this case around 90 degrees. And then once the DNA is fully single-stranded, there's no longer an increase in absorbance, so it levels off. So this is sort of a generic melting curve for any sample of double-stranded DNA. They'll always look the same. Okay, so how can you compare these melting curves for different DNA samples? What you do is you take this halfway point in the reaction. So this is what we call a TM, or the transition temperature. So at this TM value, this TM temperature, that's the point where the original double-stranded DNA molecule is now 50% double-stranded and 50% single-stranded. So that allows for, again, direct comparison between two different DNA samples that may be exhibiting a different TM value. So here's an example. Two different DNA samples. You see the shape of the curve. The melting curve is essentially the same for both, but they each exhibit a different TM value. In this case, 77, and in this case, 83. So why do they exhibit that difference? So it looks like it took a higher temperature to melt the one sample than it did the first sample, the 83 versus 77. So why did you need more energy? Why did you need more heat? Well, it's because there's probably more hydrogen bonds to be broken, meaning that there's a higher GC content. So again, the higher the TM value between any two different double-stranded DNA samples, the higher the GC. The higher the GC content, the higher the TM value. So again, there's a direct proportionality here. So here's GC content on the one axis. Here's the TM value on the other. So you can see there's a linear relationship here. So if you have a molecule of DNA that's 100% AT, meaning there's 0% GC, then your TM value is going to be around 70. If you have a DNA sample that's 100% GC, the TM value is going to be about 113 or so, up here at the top, goes off the graph. So about 113, 114. And then you have everything in between. So here, at the bottom of this linear, or this line, this graph, is that here you have predominantly AT base pairs and a little GC, but at the top, you have a high percentage of GC base pairs and a low percentage of AT. So again, it's the idea is that if you discover a new life form, all you have to do is measure the TM value of that new life form, bring that TM value to this graph, and read off and get a pretty good estimate of the relative proportion of GC base pairs in that DNA sample, in that organism's genome. And once you know the GC content, then you know the AT content. So it's a very quick and straightforward way of beginning to describe the organization of the genetic information for that particular new life form. Okay, so with those different properties, measurable properties, again, we'll refer back to them in upcoming lectures, but I just want to conclude this discussion with this quote from the Watson-Crick paper that was published in the spring of 1953, where they described their model for double-stranded DNA. So what they said is that it has not escaped our notice that the specific pairing, again, the nature of the base pairings, AT and GC, that we have postulated for our model immediately suggests a possible copying mechanism for the genetic material. So the idea of all of this discussion on the chemistry of nucleic acids, the structure of bDNA, the Watson-Crick model, and so on, is understanding structure will allow you to understand function. So again, we described 4 different functions that are associated with the genetic material: faithful replication, the ability to encode information, the ability to express that information in a cell, produce proteins, and lastly, then the ability to change or undergo a process of mutation. So all of those functions relate very directly to structure, to the structure that we've described. So Watson and Crick knew that. They're describing structure, but they said, okay, based on our structural model, we can immediately suggest a mechanism for function, and specifically the function of faithful replication. So we're going to start talking about this process of DNA replication in our next lecture.